Hi team, and welcome to this video talking about the Calvin cycle, the second part of photosynthesis. We're going to be talking a little bit about what we do with those outputs from the light reactions. That is the first part of photosynthesis, part 1. And the light reactions produce, you'll recall, quite a bit of NADPH and some ATP. You'll recall from talking about NADH in mitochondria that this NADPH is an electron carrier, and ATP is also carrying a lot of energy as well. And just as a reminder, oxygen is also produced here as a byproduct, and that is what we breathe in the air every day. We're going to use the NADPH and the ATP produced in the light reactions in the Calvin cycle to help produce sugars. So let's talk about the Calvin cycle. Since we're building sugars in the Calvin cycle, this is an example of an anabolic series of reactions. Anabolic, we're going to need to add energy, and this energy in the NADPH electron carriers and in ATP is what we're going to use to fuel the synthesis of sugars. The Calvin cycle starts when we add the gas, carbon dioxide, to a precursor molecule called RUBP. And RUBP has phosphates on both ends here. RUBP is a 5-carbon molecule. CO2 and RUBP are put together to form a 6-carbon intermediate that again has phosphates on both sides, and that reaction is catalyzed by the enzyme Rubisco. Of course, this 6-carbon molecule is going to spontaneously break down right away into 2 3-carbon molecules. This first part of the pathway is called carboxylation. We're adding CO2 onto RUBP with the help of Rubisco, and that's carboxylation. And Rubisco is actually the most abundant protein on Earth, mostly because it's pretty slow at doing this carboxylation step. Now, once we've got these 2 3-carbon molecules, we're going to take 2 ATPs, and then, of course, release 2 ADPs to produce a doubly phosphorylated molecule. And of course, we'll have 2 of those as well. Next, we're going to lose one of these phosphate groups yet again to produce a singly phosphorylated molecule, G3P. So here we're losing 2 phosphates because we've got 2 molecules here. And G3P, you'll recall, was one of our intermediates in the citric acid cycle. So in order to produce this G3P, we actually need to use NADPH and then release 2 molecules of NADP+. Now, NADPH and NADP+ are just like NADH and NAD+. It's just that they have a phosphate group on them. Now that we have these molecules of G3P, one of the G3Ps is actually going to go off into the cytosol and be processed into glucose. Now, glucose has 6 carbons, so we need 2 G3Ps ultimately to help produce glucose. And then glucose, can be stored as cellulose or starch And this is what we'll eat when we eat plants. We get much of our energy from the glucose that's stored as starch inside plants. Now, something I want to point out here is if one G3P is leaving to help form glucose, that's 3 carbons that are leaving the pathway. But up here at the beginning, we only added in 1 carbon dioxide. Now, to make this a cycle, that means we actually need to think about doing this pathway in multiples. We need to balance the equation. Let's think about starting with 3 entering CO2 molecules. That means we're going to need 3 partners of RUBP. And we're going to make 3 molecules of this 6-carbon intermediate, but it will break down into 6 of these 3-carbon phosphorylated groups. We're going to need 6 ATPs and 6 ADPs will be released when we produce 6 of these doubly phosphorylated intermediates. Then we're going to need 6 NADPHs and 6 NAD pluses will be released, along with 6 phosphate groups when we make 6 G3Ps. Now, if one G3P, 3 carbons, leaves to make glucose, well, we'll still have 5 molecules of G3P left in the pathway. So with our balanced equation, we have 3 CO2s that have entered the pathway. We have 3 carbons that are going off to make glucose. And then we're left with these 5 molecules of G3P. So we've got 5 molecules, and each one has 3 carbons. That means we have a total of 15 carbons here. What's going to happen to these 5 molecules, these 15 carbons bound up in G3P? They're actually going to go on to rebuild RUBP, our original starting molecule. Up here in RUBP, each molecule has 5 carbons. We've still got a total of 15 carbons, so that means we have 3 molecules of RUBP times 5 carbons, 15 carbons up here as well. We go from 15 carbons down here, 15 carbons up here. The rest of this pathway is just going to be rearranging or rebuilding this RUBP, ribulose bisphosphate starting molecule. And in doing that, we're going to use 3 molecules of ATP and release 3 molecules of ADP. And those phosphate groups are up here on RUBP. So that's our Calvin cycle. Let's walk through it one more time. We take in molecules of CO2 gas, and that's joined together with ribulose bisphosphate, RUBP, by the enzyme Rubisco. And then we form a 6-carbon molecule first, but it quickly breaks down into 3-carbon phosphorylated molecules. And then with the help of ATP and NADPH, we're going to produce G3P, some of which can go on to help make glucose. And this is what we eat when we eat plants. I mean, we eat proteins as well, but we're also eating this glucose, cellulose, and starch. And then the remaining G3Ps get reorganized and rebuilt to make more RUBP and start the cycle all over again. Now, the Calvin cycle is usually split into 3 stages, and we started here by talking about carboxylation. The first phase is carboxylation, where we add CO2 onto RUBP. Carboxylation, or adding CO2, and that's catalyzed by Rubisco. The second part of the process is called reduction phase, right here. And in this case, we're reducing carbons. We're going, so up here in this first phase, we've got CO2. It's fully oxidized as CO2. But down here, our carbons aren't only bonded to oxygen anymore. They've been reduced. We've reduced the carbon by making carbon-carbon bonds. But of course, if we're reducing, we're also oxidizing something. We're going to be oxidizing NADPH into NADP+, for example. Last but not least, this is the rebuilding phase where we're reorganizing these 5 G3P molecules, or 15 carbons. We're rebuilding and rearranging them into 15 carbons, but they're in a 5 carbon molecule now of ribulose bisphosphate. And that's made up so that we can start this pathway all over again with the help of Rubisco. All right, team, I hope that this helps you understand a little bit more about the Calvin cycle and the processes that are happening therein. When we add CO2, then we reduce those carbons and finally rebuild our starting molecules. And let's not forget that we lose some of our carbons out here to turn into glucose and other delicious things inside the cell. And the Calvin cycle is intimately connected with the light reactions because we're using those NADPHs and ATPs over here to integrate all of the parts of the pathway together. So bring any questions you have for me to class. See you soon.